Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to talk about positive inotropic agents. So with that, let's give it a go. So what are inotropic agents? So inotropic agents are factors that either increase or decrease the contractility of the heart. So in this video, we're going to be talking about the positive ones, and these are going to be ones that increase contractility. And the main mechanism that they're going to do is that they're all going to basically increase the amount of calcium in the cytosol. Because when you increase the amount of calcium in the cytosol, you increase the ability of the heart to act as a better pump. So you're increasing the contractility or the intrinsic ability of the heart to act as a pump. So all of these are going to increase calcium inside the cytosol. So let's take a look at a few examples. So the first one are the adrenergic agonists. So before we get into what these do, I want to give a quick review as to how calcium enters into the cell during excitation-contraction coupling and how that calcium is removed from the cytosol in order to facilitate relaxation. So let's just say you have a positive charge move down the T-tubule and hit the L-type calcium channels. This will cause a conformational change which opens the channel, allowing calcium to flow into the cell from the extracellular fluid. The calcium then can interact with the ryanodine receptors, which causes conformational changes, which allows calcium to flow from the sarcoplasmic reticulum into the cytosol. So this whole process is called calcium-induced calcium release, or CICR. So when the calcium levels increase, this allows contraction to take place. Now, in order to stop contraction, like we said, we have to remove the calcium from the cytosol. And the ways that we do that can either be in the plasma membrane, where you remove it from the cell altogether, or through the Ca pump, which will bring it back into the sarcoplasmic reticulum. So the ones on the plasma membrane are all driven by the sodium-potassium pump, which pumps 3 sodiums out, 2 potassiums in. So this then establishes a sodium electrochemical gradient, which the sodium-calcium exchanger uses to take calcium out of the cell. So the sodium-calcium exchanger is a secondary active transporter, which uses the energy released by bringing 3 sodiums into the cell in order to drive the uphill movement of calcium out of the cell. Another mechanism is going to be through the PMCA, which is a pump which pumps calcium out, protons in, by hydrolyzing ATP. And the last way, which is the most important way, is through the Ca pump, where it uses ATP in order to pump 2 calciums into the sarcoplasmic reticulum, 2 protons out, and it uses the ATP hydrolysis to drive this. So now that we know the calcium removal mechanisms and how it gets in, let's see how the beta-1 adrenergic receptor agonists are going to affect these things. So if we bring in an agonist and bind it to the receptor here, what's going to happen is it's going to cause the GS cascade. So it's a GS coupled protein receptor, or a stimulatory, and basically this leads to an increase in cyclic AMP. So the cyclic AMP is then going to activate protein kinase A. And protein kinase A does a ton of things. So the first thing is it's going to phosphorylate the L-type calcium channels. And when it phosphorylates the L-type calcium channels, this is going to increase the open probability of these channels, which is going to allow more calcium to come in. And when you allow more calcium to come in, this increases the amount of CICR. In other words, by allowing more calcium to come in through the L-type calcium channels, more calcium can interact with the ryanodine receptors, which allows calcium to flow from the sarcoplasmic reticulum into the cytosol. So this whole process is called CICR. And when we increase CICR, this increases the amount of calcium levels inside the cytosol, which therefore increases the contractility. So the next thing is going to be through the ryanodine receptors. So the ryanodine receptors, when you phosphorylate these things, is going to increase the open probability of these channels, which therefore increases the amount of calcium released from the sarcoplasmic reticulum, which therefore increases calcium levels and increases contractility. So another target is actually going to be the phospholamdin, and this is going to affect the heart rate. And how it's going to do that is when you phosphorylate phospholamdin, this is going to increase the activity of the Ca pump. And when you increase the activity of the Ca pump, you increase the speed of relaxation, which is also called the lusotropic effect. And when you do this, you decrease the duration of contraction, which therefore increases the heart rate. So another thing that the beta-1 adrenergic receptor can do is that the GS protein that you stimulated or activated can actually open L-type calcium channels, which allows more calcium to flow in, which allows an increase in calcium levels in the cytosol, which therefore increases the contractility. So the beta-1 adrenergic receptor agonists are going to increase contractility by increasing the amount of calcium in the cytosol. So another one is going to be the Digitalis derivatives. So Digitalis derivatives are molecules that can bind to the sodium-potassium pump and inactivate it. When you do this, this keeps sodium from leaving the cell, which therefore increases the sodium levels in the cytosol. And when you increase the sodium levels in the cytosol, this is actually going to inhibit the sodium-calcium exchanger. And when you do this, you basically prevent calcium from leaving the cell. And when you prevent calcium from leaving the cell, you increase calcium, which therefore means you increase contractility. So a third inotropic agent, positive inotropic agent, is going to be high extracellular calcium. So what this is going to do is it's first of all going to affect this step. So if you have a higher amount of calcium in the extracellular fluid, when the L-type calcium channels open, this means that more calcium is going to flow from the extracellular fluid into the cytosol. Another effect is it's going to affect the sodium-calcium exchanger. So it's going to inhibit the sodium-calcium exchanger or decrease the activity of it, therefore allowing more calcium to stay in the cell, which increases the amount of calcium, which increases contractility. So another positive inotropic agent is going to be low extracellular sodium. So this is going to affect the sodium-calcium exchanger again, because you have less sodium inside the extracellular fluid, which decreases the electrochemical gradient for sodium, which therefore leads to a decreased amount of energy being released by the sodium-calcium exchanger when it brings sodium in, and it therefore decreases the amount of calcium that can leave the cell. So it's decreasing the activity of this exchanger, which therefore causes less calcium to leave the cell, which therefore increases the amount of calcium in the cytosol, which increases the contractility. So in summary, we talked about how positive inotropic agents increase calcium inside the cell, and we talked about a few of them. And one of the ones that we didn't talk about, though, was the staircase phenomenon. And the reason why is because I have a video on that already in my heart physiology playlist. So I hope this video helped you understand what these things are, and I hope to see you in the next one. Thanks for watching.